All right, great. Thank you very much for the opportunity to speak to you. It's always a great experience how BioPhysical team manages to bring a crowd that's excited about one topic. One is it's definitely relevant for drug discovery and also in future. So my talk will be around cheaper, faster, smarter lead generation. And first I will introduce why a small young company like us are claiming, making such claims, and then we'll present a couple of case studies that actually demonstrated. So I'm Serghei, co-founder of CrystalsFirst, and we actually come from the group from Gerhard Kleber, from beautiful Marburg. And when the company was in formation, I did some research, and actually Gerhard's group deposited roughly 1% of the whole PDB. So a lot of structural biology work has been done there. And there was a discovery that we developed into a technology, and now we're operating as a company focused on crystallographic fragment screening and fragment-based drug discovery. I knew that Mark was going to use Henry Ford quote, and I like this one because this underlines what we are doing, have been doing for years. So if you always do what you always done, you always get what you always got. So we kind of also rethinking in a positive way, how can we enhance screenings and contribute to drug discovery. And there is no doubt and that structural biology contributed a lot to drug discovery. There are even publications around that topic that underline that. And all the tools that we are talking about are somehow based on the structural data. And in an fragment-based drug discovery setting, the typical setup for fragment screening has a kind of two steps, like a primary screen, secondary screen, then we go into structural biology. And structural biology is required for FBDD to progress because there are multitudes of binding modes that fragments can find on the surface or in pockets of proteins. And only after the biophysical screening cascade, the structural biology kicks in, and we can start iterating and designing the molecules once we have the structure going to micro nanomolar compounds. But we wouldn't be CrystalsFirst if CrystalsFirst like a slogan. And there are several problems with biophysical screenings. Pardon the chaotic images, just showcasing that combination of biophysical methods, there's a poor overlap between them. And the question is, what is a fragment hit? What do we prioritize? How much time and effort do we invest in order to validate? Because by the end of the day, we all need the structural data. So we need the binding mode. And there are publications, and it's kind of increasing, that the overlap is poor. So Uta showed us that for chemical spaces, overlap is a good thing. Poor overlap is a good thing, but in terms of prioritizing fragments here from biophysical screenings, it's difficult. So we can start investing time into validating it. And also during the formation of CrystalsFirst, there was this pivotal study from Gerhard's lab where they compared 6 biophysical assays and X-ray crystallography, screening by X-ray crystallography on a model protein with 361 compounds. And here also we had, there was poor overlap. But the intriguing part is a combination of screening methods in a cascade would have retrieved a maximum of 19 X-ray structures, maximum. And if we compare it to 71 X-ray structures, it's kind of totally different dimension. We have much more starting points. And again, we have an overlap of 0 between all 6 methods. So there are a lot of questions. What is a hit? How to prioritize? So why don't we start with a crystallographic screening fragment-based drug discovery directly? And I hope there are some crystallographers here in the audience. So feel free to type your comments. What do you think of this, of the next slide I'm going to present? So quickly, just to set a baseline, there are two ways to determine co-structures of ligands and protein. We can co-crystallize, or we can soak. So soaking works with apo proteins, and co-crystallization, we need to put the fragment and the protein into one solution. I hope they will co-crystallize, so we cross-crystallize. And soaking is actually a more scalable method because we can produce a lot of apoproteins and soak more compounds. We collect data, we derive the electron density, and then we model the protein and the ligand. All right, and CrystalsFirst has a unique technology for soaking. But before we dive into that, the critical factors for protein crystallography. So I hope there are some crystallographers or people who experience that. So one critical factor is actually a moon phase. So you have to align your experiments when, depending on the protein, which moon phase works best. We make the experience that protein crystallize much better in spring season. And of course, pay attention to weather. And of course, if you are a magician and you have voodoo skills, you will be a great crystallographer. Of course, I'm only joking, but it kind of emphasizes the jokes in protein crystallography, because it's a tedious process. It's not even a process. There's a lot of trial and error. And any technology that would contribute to solving problem, accelerating the process, making more robust, is very welcome. All right, so coming back to the question, why don't we start directly on screening on crystals? Because it's the most sensitive method compared to other methods. It offers the hit and the context of the binding event. It's not only binary question, it offers the context, and we can start applying all those efficient tools for modeling. But of course, there are problems with crystals soaking process. And what we hear a lot in discussion is that the most common phenomenon is no structure or empty structure, which actually can be solved with technologies that we apply. Of course, speed is misaligned with medicinal chemistry. The resolution is an issue for fragment-based drug discovery, and not having this structure actually deaccelerates the whole process of drug discovery. And we define what is conventional soaking. Conventional soaking is driven by trial and error. It is, so protein crystals are very sensitive and their behavior cannot be predicted. And what is also important, if we have many compounds, the solubility issue actually is a problem. So we get empty structures or like no structures from co-crystallization at all. So this is kind of the industry standard. We have sensitive crystals, there is no process how to treat them. But with CrystalsFirst, we have actually technology that solves this problem and therefore accelerates the process from the crystal to 3D model by a factor up to 10. So going from trial and error to having a process is like 0 to 1. And we have also a P for this technology we applied for. And just to show you, we've been operating 3 years and we applied to multiple targets, the technology. There are targets I can talk about. There are also targets I cannot talk about, but it's target agnostic. It's proven to work. I think we're heading to 30 targets right now. And we deliver success rates of 30%, which means we don't lose protein crystals. We work with higher concentrations of ligands. That's why the success rates are just higher. So better structures faster, high performance soaking systems. We stabilize protein crystal native states. It's not cross-linking. It's not cross-linking. We work with high concentrations of ligands. It's important to determine the co-structures. So here's an example of two compounds bound to the surface of a protein, same binding site, same compound. And Unfortunately, the industry standard for soaking, from discussions, is actually low millimolar concentration for fragment. And for crystallographers, this electron density actually noise for good crystallographers. Only at 9 milli we have one compound with resolved electron density, but the second still noisy. With 90 milli we have well resolved electron density. And with 100 millimolar concentrations, we see that we have much higher success rates because of this phenomenon. It's not a functional assay; it's X-ray crystallography. We need high concentrations, and optimally at each monomer there is one compound. We soak for longer periods of time, and we improve ligand soluble applicability. So to compare, SMARTSOAK stabilizes crystals. Their behavior becomes more predictable. It becomes actually predictable once they're stabilized. We don't need trial and error optimize when we soak compounds, and we use higher soaking concentrations. And as a young company, we went out with this approach, with this technology. We talked to the industry 4 years ago, and of course, the industry response was, we need affinity, we need larger compounds, etc. But we persisted, created case studies, and I'm excited to present one of the next case studies here. And if there are people from Boehringer on this webinar, on this symposium, thank you for this publication. It's it's actual application of our approach on RAS proteins. So they call it X-ray first. They put structure biology first over affinity and optimize compounds based on that. And they also discussed that medicinal chemistry might be misguided by decision making that are rooted only in affinity. So it's a kind of a great publication that emphasizes this structure-guided approach. Now I'm switching to case studies. I have 3 case studies I have brought to this symposium. So the first one is when we started the collaboration with Analyticon. Also, we work with Analyticon very closely. They have a very interesting chemical space, NP-like compounds, but also the mix of sp2 and sp3 is very attractive. The compounds were screened directly on ENDOTHIAPEPSIN. This is a rare example of emerging process. So the compounds are bound in the active site and kind of very complementary. And once they were merged, the affinity jumped from millimolar, which is only for fragments, to low micromolar, and there was very good resolution. And we see that's the strength of structural biology. We see that the expected product is not present, something different, and the chemistry is figuring out how to optimize the synthesis so we have the required product. And the other case study was also performed with Analyticon's compounds, where we screened on a kinase. So the idea was to identify a new chemical space, untapped chemical space for kinases. And we screened an sp3 rich library there in order to identify new ATP pocket binders. So the process is very straightforward to crystallize, stabilize per SMART. So this is very efficient. It's only 24 hours. We selected the fragments, 87, and we find them. We had very good resolution, 27 compounds, a high hit rate, and an sp3 fraction. This is interesting for models. So for me as a model, it was, wow, we have in the pocket of in ATP pocket with compounds in the primary screen, they have a high sp3 fraction on average. And this is interesting because it's a different chemical space. But again, we have a crystallographic fragment screen, and we have, of course, a cluster of binders in the ATP pocket, but we have also kind of a clusters on the surface. And the question is, are those sites functional? And probably they are, because compared to this publication, there are cooperative or allosteric sites on the surface of the ATP, and 4 cluster, like the compounds address 4 out of 5 of those functional sites. So they are probably not irrelevant, but we didn't pursue to test the allosteric inhibition by those compounds, because the focus was to compare the chemical spaces. So typical hinge binders, of course, are flat, and we have like a primary hit, totally different chemistry. It can contribute just to develop ideas that can determine selectivity and also AP. And on the protein side, many things are happening. So this is kind of that addresses one question from the prior talk, like the dynamics of the protein. We see it when compounds bind, they actually move things around, and they move not only solvent but also the protein, of course. And even for small compounds, something is happening on the protein side. But like prediction of binding, nobody would have. Like, we were surprised that fluoride change interaction is present here, and there's a lot of movement here. And when we see it, we can select what structures we actually use for docking in combination with the technology that Marcus talked about. But back in the days, I just did a quick follow-up study, SRI by catalog, what is off the shelf. I selected 20 compounds just for docking, scoring, prioritizing, visual inspection. And we tested 20 in a functional assay, competitive assay. I had a success rate of 30%. 6 were in micromolar range, which is great. If you look at the ligand efficiency, I have this bar from the web, from the software, but there must be a ligand efficiency here on this side. So 1 is close to 0.3, this is interesting. And when like rapidly we got the crystal structure from this compound, and the second cycle can now begin. But like starting from very rich data from primary crystallographic screen, and then using computational methods and select the data, what can be used, what kind of chemical space we want to address. It was a very efficient process. It's a matter of weeks, actually. If things are aligned properly, it's a matter of weeks to go from target to active compounds in micromolar range. And this is kind of what we do differently. We don't use affinity data in the beginning. In this case, we didn't use it also. We just tested the primary hit, the fragment, and the affinity was like not really great, so the fitting didn't work nicely, but it was in a millimolar range. Okay, but after the case study, it inspired us. Okay, we have great results. Let's go big. Let's explore chemical spaces. And this is what we did. We sat down with ChemSpace and BIOPHYSICAL IT, and also with Professor Kleber, and discuss. Okay, we want to apply it on with this new method, and make this fragment-to-hit step even more efficient. And we had the privilege that we had a range of structures to select from, and we selected 4 starting points. These are crystal structures of the fragments bound to the hinge. And Raphael has set up, and team at BIOPHYSICAL has set up a workflow, how we start with fragments, again, chosen. So this is an important point to have multiple starting points, multiple crystal structures. It can provide the entry point to the chemical spaces more efficiently, more effectively. So they set up this chemical space docking approach. There are many details. So here's the workflow. I think the best idea is to reach out to them and have a discussion. The important part, we didn't use any affinity data. We just used host structures, and also there was a lead-like filter clustering process, of course. and 106 compounds were selected for synthesis. And there are, of course, 4 clusters of those compounds and the follow-ups. So quick overview. Sometimes there are solubility issues for measurements in the functional assay. This is not new, but we could measure all of them. The fragment here was low milli. The follow-up cluster stayed in the millimolar range. For this compound, we had a very weak fragment, actually, over 50 millimolar. But the best follow-up was around 140 micro. We did also a follow-up measurements for validation with different protein concentration. The value is actually much stronger. So we have, like, from nothing, like millimolar, just a structure. In the functional assay, it's not inhibiting, but we have kind of a boost in affinity just by navigating chemical space and selecting compounds for synthesis. And of course, I did mention it explicitly, but as the compounds were synthesized through chem space and enamine. For this cluster, we had 3 millimolar affinity for the fragment. The best follow-up compound was 86 or 30 micro. Again, we have a boost in affinity based on the structure. And for this series, the fragment was 6 milli, and the best follow-up was 1-digit micro. It's a huge boost in affinity. And this is what we see when we start with a fragment. It doesn't have to have a high affinity. Just by having this structure can provide the data that is required for the tools to explore the chemical space and select the most promising ones. All right, good. And we selected 12 most active compounds for co-crystallization. The first batch was started last week, and we collected the data on Tuesday this week, actually, from the 12 7-produced co-crystals, and we have determined co-structures. I'm going to present quickly. So let me move this bar here. So if you look at the 5 hits I have selected from the E9Q cluster, if you have a look, then you see that especially this one is very interesting. It has a good ligand efficiency and high affinity, but it's still very small. It's actually still a fragment, I would say. But this is the second best compound from the whole project. All right, so these are the 5 structures from this cluster, from this follow-up compounds. And as you see, the protein is moving, so it's not static. And I think this is important to consider, that upon binding, there are some use effective disease, the glycerol rich loop. You don't see the details, but things are moving around, and this is what we observe also. And I think this information is important to have if you start focused lead series. So the binding mode of the fragment is like this. We have hinge interaction, donor acceptor, also to threonine, but this is the amino acid. The side chain is flipping, it's moving around. Of course, we have water molecules. Water molecules often disregarded in many tools because of the complexity, but like just having this information from structural data contributes or has contributed to successful drug discovery programs that were using computational methods and structural biology extensively. Since this is very fresh data, we could have a look only at the overall performance, and the performance is actually great by the tool. So huge. So hats off for also BIOSOF IT. The tools and the evaluation of it kind of reflects the whole idea. We use the fragment as a template, and this is the predicted binding. Of course, this doesn't move, but this here we observe movement. And the question is, do we see the same in the crystal structure? And yes, we see the same in the crystal structure. The binding mode is conserved. This is what we observe. those stories about flipping fragments, we don't observe it or haven't observed so far with our programs. But this tail is moving around. Of course, most of them actually are very well in alignment with the results, with the virtual screening results. So the overall, we combine top expertise from BIOS of IT. It was only 3 weeks to us do the chemical space talking. We started with 4 structures of fragments, selected 106 compounds for synthesis. The synthesis also 3 weeks, success rate 87%, 93 synthesized. We tested in an assay, 75, 25 were active. Our co-crystallized 5 structures today, more to come, also 3 weeks. So it's kind of very fast process. The success rate, in order to there might be discussion, what is the success rate? But if we start with 106 and have 25 actives, it's already 24%. If we start with 93, it's 27. And if we look only at the tested compounds in the functional assay, we have a success rate of 33. So the success rate is around 30% overall. All right, and to summarize, what is smarter, faster, and cheaper? So I would advise to start with crystals, fragment-based drug discovery. The affinity discussion is just probably not driving the success, but being more pragmatic and having structural data is important. Multiple starting points, we see again, having a wide range of starting points contributes to success. And like in the case where I just presented, we have a success rate of 30%, and we still are in a fragment-like range. So it's kind of very exciting, very encouraging again to continue develop this paradigm. One of them, we have a boost in affinity. This is what we observe. It's not a surprise for us, going from milli to micro just from one iteration cycle. All right, we see actives, non-actives. We can derive structure-activity relationships. What is faster? Like if you do it, the net calculation is 9 weeks, like going from fragment structure to a hit, with not just a hit, but we observe affinity in a functional assay. We're inhibiting the protein. All right, crystal structure. We started with a crystal structure. We end with a crystal structure. The next iteration cycle will come, and we start with multiple horses in the race. It's cheaper. We purchase compounds just to build an SAR and structural biology, and one medicinal chemist actually is empowered to run several projects at the same time and very efficiently bring it to lead optimization stage. So I would like to, I see I have 1 minute left. I would like to kind of close the whole discussion in, I know we're a young company, but what we observed is that what is kind of the paradigm in the industry to have this screening process, high throughput screening, also fragment-based lead discovery follows this, that we start with a large set, narrow down, go into structural biology, start iterating. And there is a lot of noise. What kind of I propose is that we start with a crystallographic fragment screen. We don't have an affinity discussion in the beginning. You might run it a parallel, independent from it, but starting from the structural data, we actually expand here, visualize this horizontally, radically into chemical spaces, virtually narrow down to most promising ones, kind of expand the screening hypothesis and utilize biophysics later. But the interesting part with this is also that it's a two-way road. So we can, we will have here all again the structure, and we can jump here and there with computational methods much more efficient. So thank you for your attention. I would like to. Okay, yeah, I see. Thanks. Thank you for your attention. I would like to thank for all the collaborators, and especially also Daisy from Hamburg. They are very good partners. When we need to collect data, we get the slots, but we have also other partners, 5 different synchrotrons we collaborate with. So I would like to thank personally Gerd Kleber and Gerhard Müller, who supported focus our activities on the real problems in drug discovery, and also the whole team, and especially Yanis, who is kind of the genius in the lab, who makes many things possible in a very fast way. So big thanks to CrystalsFirst at this, also from here, from the symposium.